Translation is the process of protein synthesis in which the genetic information encoded in the mRNA is translated into a sequence of amino acids in a polypeptide chain. The process of translation not only includes mRNA, but also the ribosome. The ribosome is consisted of two parts. We have the large and small subunit. These subunits consist of RNA molecules and proteins that are then used in translation. The small subunit is the one that binds to the mRNA, whereas the larger subunit has binding sites for tRNAs, which complement the mRNA to catalyze peptide bonds between these amino acids. To look further into mRNA molecules varies. Only certain genes in a genome are expressed due to cell specialism and environmental factors. Therefore, not all genes are translated. The genetic code plays an immense role in translation. Consisted of codons, which are a triplet of bases which encode for particular amino acids. There are 4 bases, hence we can conclude that there are 64 different codon combinations. These codons can be translated into 20 different amino acids based on the amino acids that are carried by the transfer RNA. In translation, codons always start at the start codon, usually being AUG. Therefore, the first amino acid is always methionine, whereas it terminates with the stop codon. The stop codon no longer adds an amino acid to the peptide bond and instead causes the release of the polypeptide. In tRNA, the anticodons are complementary to the codons on the mRNA. These different codons can translate for the same amino acid. Therefore, the genetic code can be concluded as degenerate. The amino acids conducted from the tRNA form a peptide bind together. Let's look closely at the process. Firstly, the difference between translation in prokaryotes and eukaryotes is that in prokaryotes, the translation occurs immediately after transcription. The ribosomes in prokaryotic cells can be adjacent to the chromosomes, whereas in the eukaryotes, it needs to be relocated from the nucleus, then taken out to the cytoplasm through our nuclear membrane, hence elongating the process. First step occurs between the mRNA and the small unit of the ribosome. The mRNA binds to the small unit of the ribosome. The small subunit then moves along our mRNA molecule in the 5-3 direction until it reaches the start codon, usually being AUG. tRNA consists of a 5-3' direction. It consists of 3 different loops that work on different parts of the mRNA. Loop 2 withholds the complementary codon. tRNA with methionine, the amino acid attached to it, is complementary to the start codon, which is UAC, and this then binds to the P site of our small unit ribosome. Thus, the large subunit of the ribosome then binds onto the tRNA and our small subunit. The next step is elongation. As we can see, the large subunit of the ribosome consists of an A site, P site, and E site. The A site is the acceptor site, the P site the peptidyl site, and lastly the E site is the exit site. Second tRNA, with a new amino acid attached to it, which is complementary to our second codon on our mRNA molecule, then binds to the A site. tRNA in the P site with the amino acid is then transferred to the amino acid on the A site, where they form a new peptide bond through Candencin reaction. The ribosome then moves one codon along our mRNA molecule in the same 5-3 direction. Hence, the tRNA in the P site is then moved to the E site and released from the ribosome, whereas the tRNA in our A site is now moved into the P site. The process begins again, and another tRNA, complementary to our next codon on the mRNA, binds to the A site. Hence, our growing polypeptide increases as more tRNAs are binded to the ribosome. Then, this process is continually done until our stop codon is reached. Thus, when this stop codon is reached, we move into termination. Termination is caused by a release factor that attaches to the A site and thus releases the polypeptide chain. The ribosome is then disassembled for reuse later on in translation. Once the polypeptide chains that are now proteins are released, depending on the type of ribosome that has synthesized the proteins, they have different locations. Those synthesized by bound ribosomes round to the endoplasmic reticulum ER are secreted from the cell or used in lysosomes, whereas those synthesized by free ribosomes will be then used within the cell. There are 4 different levels of structures within the proteins. The first level is the primary structure, and this is the order of the amino acids of which the protein is composed of. These are formed by the covalent peptide bonds that form within translation. Secondary structure is consisted of these chain of amino acids, which were the primary structure. The primary structure can be conveyed as the subsequent level of structure needed to form all other structures within the protein. But the secondary structure is held by these hydrogen bonds between our non-adjacent amine and carboxylic groups. The hydrogen bonds give a stability to the structure. The tertiary structure is when the polypeptides then fold to form a complex 3D shape. And these are caused by interactions between our R groups, which are the hydrogen bonds, the ionic bonds, and hydrophobic interactions, to name a few. The structure is important for the function of specific active sites within these enzymes. Quaternary structures is when multiple of these polypeptides groups are put together and is usually an inorganic compound that involves a protein within a group. An example of this includes the hem group and hemoglobin, and these are both fibrous and global proteins together. Thank you.